Alright, so now we're going to take a look at how protein synthesis is regulated, albeit to a very abbreviated level, because this is a huge topic. But we're going to take a look at some of the places where gene expression can be influenced by factors either interior or exterior to the cell. In prokaryotes, and again, so you remember prokaryotes are things like bacteria, things that are relatively simple, single chromosome, no nucleus, not much in the way of organelles, that kind of thing, right? So relatively simple. Their DNA is actually organized into what are called operons. And so operons are like functional units of genes that include the gene itself, as well as some regulatory types of things that go along with the gene. So with the operon comes a regulatory gene, and that's the one that kind of influences how other genes are transcribed or not. There is a promoter region, which we saw before with transcription. The promoter region is where RNA polymerase knows where to bond in order to start transcribing. We then have an operator, which is kind of like a roadblock that either allows RNA polymerase to access the promoter or not. And then we also have the structural genes, and these are the ones that we actually want to transcribe and eventually translate and turn into protein. So regulatory gene, promoter, operator, and however many structural genes are in a line after that in order to make the protein or proteins needed for that prokaryotic cell. When we look at something like E. coli, so E. coli is Escherichia coli, it's a common intestinal bacterium of mammals. E. coli bacteria, since they live in the intestines of mammals, are kind of at the mercy of whatever their host eats. And so whatever their host eats is, of course, what they're going to eat. They have to be able to metabolize just about any type of food that might happen to come along. And so if they're living inside of a newborn mammal, for example, the primary food of those newborn mammals is going to be milk, and the main carbohydrate in that milk is going to be lactose. Right? So lactose, as we know, is a combination of glucose and galactose, but to break down lactose, there has to be the presence of an enzyme called lactase. So in E. coli bacteria, one of the most well-studied operons or collections of genes are those that are used by E. coli in order to make lactase to break down lactose and digest it. Okay, so we call this the lac operon. So again, there's going to be a promoter region, there's going to be an operator region, regulatory genes, and in this case, 3 different structural genes which allow for the production of eventually lactase. Right, so this picture up here is a pretty good one. It shows us the actual lac operon itself. This is part of the bacterial chromosome, of which, of course, they only have one. The DNA is here. The regulatory genes, promoter, operator, and then 3 structural genes that are all back to back to back to allow for the production of lactase. Right, so in this case here, if lactose is not present, RNA polymerase bonds to the promoter, but now there's a block here that prevents the RNA polymerase from reading down the DNA and eventually transcribing those particular structural genes. If lactose is not present, there's no reason to make lactase. To do so would be a waste of energy, and so this repressor protein prevents lactase from being produced when it's not needed. On the other hand, if lactose is present, that lactose bonds to that blocker, that repressor protein. So when RNA polymerase bonds to the promoter, it can go through the operator, the roadblock is not in place, and eventually RNA polymerase can then transcribe the 3 structural genes into RNA, and then RNA can eventually be turned into protein. So it's a matter of whether or not lactose is present as to whether or not the bacteria can actually produce lactase. Right? So in eukaryotes, it's a much more complex kind of a thing because we have a lot more different possible control points. We also do not have the genes organized into operons like we saw before with the prokaryotes. It's a much different kind of a situation. So the picture on the right here actually shows us a very schematic, idealized view of how gene expression is controlled in some of the points along the way in transcription and translation. So the way this picture is set up is it's set up like a pipe. And the pipe in this case has several different control valves. In the case of the pipe, though, the control valves are controlling information flow, not water flow. So it's controlling flow of information. Right? So if the valve is turned on, we go to the next step. If the valve is turned off, we're stuck at that particular step. Okay, so all the way back here at the level of the nucleus, there are control valves to allow for DNA to be packaged up more tightly and therefore not read. Other kinds of changes to DNA which might prevent it from being transcribed, and of course if it's never transcribed, it's never going to be translated. If this valve is opened, so if DNA is unpacked enough to access the genes, the next control valve we have is transcription. And you'll notice how this valve is different from all the other ones in this picture, and that's because the transcription valve is special. There is more gene expression level control that occurs at the level of transcription than there is at any other point in the entire process. So transcription is a very important point where gene expression can be controlled. All right, we can then have the addition of the cap and tail. We can also have the splicing control valve. Both of these two are involved in processing mRNA before it's allowed to leave the nucleus. So if these two things don't happen, mRNA will not leave the nucleus, and therefore you won't have any translation. We have a control valve to allow the mRNA, the processed mRNA, to go through the nuclear envelope. We have a control valve that we can use to break down mRNA if we need to. So if we actually go to the point where we make mRNA but then don't need it, we can break it down by using a set of enzymes on the cytoplasm. Of course, that's fairly wasteful, and cells would prefer to do that if they can possibly help it. But in some cases, by the time you make a protein, the protein might not be required anymore, and breaking it down would actually save on energy as opposed to just keeping it around doing nothing. We also have a translation valve, so all the stuff we talked about in the translation step can be regulated too. We have a protein modification valve, so all the stuff that takes place not only in the rough ER but also the Golgi apparatus, that can be regulated very precisely as well to give a very fine degree of control over translation. And then we also have the possible breakdown of the protein itself, and that might be the least desirable option, to spend a bunch of energy and resource making a protein which you then break down. But in some cases, it might actually be more energetically beneficial to do that rather than try to use a protein or have too much of a protein that you don't really need. Right, so that's another type of control we see. And so this picture on the right over here, this figure shows us, okay, packaging DNA, transcribing DNA to RNA, processing RNA with caps and tails and splicing it, allowing it to go out to the cytoplasm, breaking it down possibly, or allowing it to be translated, modified, and or broken down after that. So there's a lot of different places along the way here where we can actually control how transcription and translation work. All right, the last section of this chapter is going to deal with mutations in DNA. We're going to talk about how accurate DNA replication is, and it is actually a very accurate process, as well as gene expression. They're very accurate processes. Mistakes do occur. So when DNA is copied or when DNA is transcribed and translated, there's a very, very, very low likelihood of a mistake being made. And most of the time, even when mistakes do happen, they get fixed by the cell. So in very, very rare instances, we do get cases where mistakes happen, they don't get fixed, and they actually get through the quality control process with the cells have to keep their DNA intact. Okay, so sometimes the mutations are very severe, sometimes they're not. Sometimes mutations are a single letter change or deletion or addition. Sometimes they're more substantial than that. The actual effect of the mutation depends on how much of the gene is actually affected and how it's affected. Okay, so mutations can be, in many cases, neutral, not really have much of a noticeable effect at all. In some cases, mutations can be very detrimental, very harmful, maybe even lethal. And in a few rare cases, mutations can be beneficial by providing some new trait or behavior or something like that to an organism, thereby increasing its fitness. Right? So one type of mutation that we see in organisms can be called a point mutation. And this one's also called sometimes a substitution mutation, where you take one letter and switch it out for another one. You change an A to a T, or a G to a C, or an A to a G. And depending on what effect that has will determine how substantial, how significant, how important the mutation is to the overall fitness of the organism. Now, in many cases, back at the beginning of the last section, we showed you how the genetic code table is set up. And we talked about how the genetic code is redundant. So even if you mix in the wrong letter from time to time in terms of bases, you might still get the same amino acid. You might still get the very same protein, even though the gene is officially changed or maybe even considered broken in a way. So point mutations, because the genetic code has a degree of redundancy built into it, point mutations tend to not be super severe. Doesn't mean they can't be, because sometimes they are, but most of the time mutations that occur are going to occur in point mutations can be somewhat neutral. Right? In the case of sickle cell anemia, this one very much is not a neutral mutation. So in this case, we have just a very, very tiny set of the bases that we see in the protein hemoglobin, right, to produce the protein hemoglobin. So those bases read like this. We have bases that code for leucine, and then threonine, and then proline, and then glutamate, and then lysine, and then serine. In some individuals, there's a change here from glutamate to valine. So instead of two of these guys, we have a valine and a glutamate. And by changing just that one base, that affects the protein function completely. And if you have normal hemoglobin, your blood carries oxygen, your blood does a pretty good job of distributing it to where it needs to go in the body, also carries CO2, and you don't really have to worry too much. If you have sickle cell anemia, especially depending on how many versions of it you get from your parents, it can be lethal relatively early on in life. And in many cases, even if you have only one copy of sickle cell, it's debilitating enough that it becomes very difficult to lead a healthy, normal life. So that's a very severe change. Just to give you some idea of that, those that have sickle-shaped cells, red blood cells, some of the issues they run into are the early breakdown of red blood cells, the fact that these blood cells, because of their shape, the sickle shape, tend to stick together. They clog blood vessels, they cause swelling and pain, those kinds of things. It also overworks the spleen. The spleen is responsible for recycling a lot of the parts of red blood cells back into circulation. We also have these effects. So we have weakness and anemia, heart failure, pain, fever, brain damage, other organ damage, and spleen damage. And these things all lead to these effects. What's not pictured here is death in some cases. So just by changing that one letter out, by changing out this, this A to a T, we go from glutamate to valine, which means we go from having cells that have the round shape to cells that have this shape. And when your cells have this shape, there's a whole set of challenges you run into as a result. And we'll talk more about hemoglobin, sickle cell anemia, and actually malaria later on in a different chapter. We'll see that one again. All right, the next two types of mutations are very much similar to each other in terms of their effect. One of them is an insertion mutation where we add in one or more bases. The second one is a deletion mutation where we take out one or more bases. Now, these two mutations tend to have a more important effect on phenotype or physical appearance or behavior than do the ones that we just saw. Right? So with insertion or deletion mutations, the problem is we're going to leave out one or more bases or add in one or more extra bases. And that can, again, have varying levels on the outcome in the organism. If it's a very critical gene, there's a chance that you might actually have an overproduction of a protein or the wrong protein, and that's going to lead to some very severe consequences for the individuals that have the mutations. Now, the issue with this is the fact that if you add in one base or take out one base or add in two bases or take out two bases or 5 or 7 or 8 or whatever, as long as it's not a multiple of 3, it can be very, very common to have very severe mutations. And what is more, if you start deleting or adding in extra letters that are not multiples of 3, that throws off everything downstream for what was supposed to be there before. It actually throws off what's called the reading frame, which is how our ribosomes scan down the mRNA, read codons, and plug in appropriate amino acids. And that's a very, very dangerous situation to have mutations that actually result in those kinds of changes because it completely and utterly changes the shape and function of the protein. Inversion mutations are where a piece of DNA is actually taken out of the chromosome, flipped around 180 degrees, and stuck back in again. Right? Now, you might think that sounds pretty bad, but it turns out that if you move the entire gene out of the chromosome, flip it around, stick it back in, it's perfectly fine. It functions just like it was supposed to function, even though it's been flipped around 180 degrees and stuck back in again. If, however, you only take out half the gene, flip that around and put it back in, that's going to lead to the breaking of the gene and therefore some sort of physical consequence potentially that you might see in the individual itself. Okay, so by flipping it around, it's really important to make sure you do that with the entire thing. And so again, none of these things are intentional. None of them are done on purpose or consciously. They just sort of happen. And if it happens that a mutation is one that affects the nose, then that's the way it's going to be. Or if it's going to affect some other part of the body, that's the way it's going to be. Right? So that's the issue we run into with these types of mutations. They can be very severe if only part of the gene is taken out and put back in. Translocation mutations are kind of similar in that way, in that if you take out part of a gene and move it, it actually breaks the gene. You put one gene that was on chromosome 13, move half of it to 17, leave the other half on 13, that gene is now effectively broken and it's not going to work anymore. If, however, you move the entire gene, that might actually not be that bad. So if you move the entire gene from 13 to, say, 15, that might not actually be a problem at all. So translocations, again, depend on the specific set of circumstances that we see as far as what kinds of effect it's going to have on the organism and our fitness. All right, so that's the end of this chapter. We're going to cut it off here, and the next chapter we're going to see is going to be on mitosis and cell division.